Hello, today we will be talking about the blue bottle experiment, which is a redox reaction example. In this video, we're going to be talking about our theory, some kind of keywords like materials or key aspects of chemistry up inside the experiment. Then we're going to be talking about the fundament, why we chose that experiment, and finally, we will be talking with the theory. At last, not and regardless, not to mention that we will be explaining how this works and the results shown.
Let's go with the theory. Oxidation reduction, redox, reaction is any chemical reaction in which electrons are transferred between two participating reactants. The transfer is identified by noticing the change of the oxidation number of an ion, atom, or molecule, and see if it is losing or gaining an electron. Reduction. In any class of chemical reaction, the reduction is a process in which the number of electrons associated with an atom or group of it is increased when it accumulates electrons during a reaction. Oxidation. In reaction terms, the oxidation is the process of transfer of electrons in which a reactant loses some of them during a reaction. Oxidation state, also called as oxidation number, is the total number of electrons that an atom either gain or lose, so a chemical bond can be formed with another atom. It can be seen on the periodic table of elements.
The blue bottle was a chosen experiment due to its surprising color change reaction from blue to transparent when the bottle is shaken. Apparently, during the experiment, the sugar is hydrolyzed to break the hydroxidic bond between the glucose and the fructose. So there will be a node glucose that reacts with NaOH to produce glucose enolate. Then the enolate should make a redox reaction with the methylene blue. The glucose will be oxidized by the oxygen of the air in the bottle to form gluconic acid represented in sodium gluconate, while the methylene blue is reduced to colorless leucomethylene blue. According to this, we want to know how it visually represents the oxidation and the reduction of the substance. Also, we want to identify why the surface of the substance is blue after the shake. In addition, the bottle is going to be shaken more than one time to see if the results of the experiment are the same or varies.
Well, the method for this experiment is to first have a beaker and a Erlenmeyer flask. Next, in the beaker, we put like 4 teaspoons of sugar. Next, we add like 25 30 milliliters of water. Next, we put lemon into the solution to make acid glycolysis. Next, we heat the beaker for a couple minutes, and while the beaker is heating, we mixing and to make faster the process. Next, we add like 2 or 3 teaspoons of sugar of sodium hydroxide and put more water. Next, we drop 5 drops of methylene blue into the solution to oxide the solution. Next, we pour the solution into an Erlenmeyer flask with a cork, and we put the cork, and to the end we shake it and see the results.
As expected, as we mentioned in the fundament, in the first try, when the Erlenmeyer flask was shaked, the substance was oxidized, showing a light blue color, meaning that was the methylene blue. And then after 3, 4, maximum 5 seconds, we can notice how that oxidation was reduced then to a leucomethylene blue. And when we repeated to the second time, things were different. Regardless, the color was still a light blue. The time was a little bit slower. from passing from the oxidation to the reduction state, not from 3 seconds, but from 8 probably. And after repeating 4 or 5 times, the color was not a light blue anymore, it was a dark blue, and the time to pass from the oxidative state to the reductive was even more time, probably 30 seconds. And last, we after 10 tries, the color was not yet a blue, but it was a purple, and not a light purple, not it was its opposite, the dark purple. This could happen because well, and also I forgot to mention that also the well, the time to pass from the methylene blue, that in this case wasn't methylene blue anymore, but to the transparent state was more than a minute. And every time we shake it and we measure the well, yeah, and we took the time, it was even getting more and more time. For this, we have these three thinkings. The first one is that maybe since the bottle was before during the method being hydrolyzed, but has it hydrolyzation, but well, also by adding heat with a heat system, maybe the maybe the substance was getting cooler, so that means the glycosidic bond is that was broken joins again, and the glucose became again a saccharose, making the join by media the glycosidic bond of the glucose and the fructose. Remembering that in this case our experiment was doing it was made with the acid hydrolyzation and adding some sugar instead of glucose. That was why the importance of the hydrolyzation from the heating system. And well, we thought that we are thinking that the more cold it is, the substance, the effect is going to be loose. And in some point after some shakes, probably more than 30, the this type of redox reaction is going to lose its type of luxury of becoming from one substance to another.